Welcome everyone to our seminar this noontime. This is the regularly scheduled grand rounds for the Department of Environmental Health Sciences. And of course, all of you know that April is EHS month in celebrating the school's 100th anniversary. So I'm especially pleased to co-sponsor this seminar with the department. So Cigarette Restitution Fund sponsors 12 lectures a year. We broadcast them, we record them, we post them on the web, and so it makes them for a nice combination when our topics overlap. So today I've invited Karen Stefanos to join us. She's a new faculty member in the School of Medicine, Department of Pathology. And she actually received an award from the Prostate Cancer Foundation, which really gave her a start with her topic for today. So she completed her PhD in cellular biomedicine. There. In the School of Medicine. And she worked with Bill Isaacs and Angelo DiMarzo, so no doubt you'll hear some of their work. And you know too from the title that we couldn't leave them out. So Karen promised me though, or suggested that we may have some controversy, in her presentation of the microbiome, knowing that this department has a major theme in that area. So would you welcome Karen Stefanos?
Thank you very much, and I'm so happy to see so many familiar faces in the audience, as well as some faces that I don't recognize and haven't seen before. So I really appreciate the opportunity to come over here to School of Public Health, where they videotape you while you're giving a presentation, to tell you a little bit about the work that we're doing on a broad number of themes, but largely speaking on infections, which is the major theme of my lab, studying prostate infections and how those could relate to prostate cancer. PHIP, a dietary carcinogen, which I will tell you some about because I know there's a large interest in that here with some members of the audience. And this is an outline of the talk that I'm going to give today. So I'm going to start out by telling you broadly about prostate inflammation. Can you hear me better? Wonderful, thank you. All right, can you do me a favor? If it tends to slide and you can't hear me, just wave at me and I'll, yeah, okay, thanks. All right, so I'm going to start out broadly talking about infections and inflammation in the prostate and try to give you a little bit of background about why we are studying this and why we think it's related to prostate cancer. And then I'll move on into talking about PHIP as a dietary carcinogen. When Norma and I were initially discussing this seminar, she had said that she was looking for someone to talk about diet and prostate cancer, and I was like, well, I don't really study diet and prostate cancer, but I do have, a paper that just came out on PHIP, and so I'd be glad to discuss that in the context of infections. And then a few days later, she sent me another email that said, and by the way, the department is very interested in the microbiome, and I see that you do, you're moving into a microbiome related field. And so that's what I was telling Norma about, where at the very end, if you stay awake all the way through till the end, I will talk about some very recent work that we're doing in the microbiome field. And the controversy is that it's urinary tract microbiome, which up until this point, people didn't even believe existed. So we'll get to that as the second part of the talk. And then I have this slide in here just to remind me to acknowledge up front, and Norma already alluded to this, that I list them as collaborators, but they're certainly also mentors, both Angelo DiMarzo and Bill Nelson. I can't give a talk about PHIP without mentioning that the two of these people, who I will highlight in the next several slides, have been working on this hypothesis and these ideas for many years before I was even a graduate student, and really were the ones that brought the PHIP model, the PHIP-RAT model of prostate cancer that I'll be telling you about here to Hopkins. So I will show you a lot of histology pictures just because I like to show pictures of histology along with what I'm talking about. I think it's a nice way to just show you what I'm thinking. So this is what normal prostate looks like, right? So there's two epithelial cell layers. There's these luminal epithelial cells, which are responsible for this prostate secretions that are secreted into the lumen. And then there's this basal cell layer that lies underneath the luminal epithelial layer. And then there's this fibromuscular stroma that comprises what normal prostate looks like. And I'm absolutely delighted that Dr. Coffey is here today, because every time I show this next slide, I talk about Dr. Coffey and say that normal prostate is not what you see in the adult man. When you look at prostates that have been removed for prostate cancer, for benign prostatic hyperplasia, for autopsies, prostates in the adult man, the prostate actually looks like this, which what Don Coffey refers to as a trash dump, which is the best explanation that I can possibly come up with for what this actually looks like. The architecture looks completely different. More often than not, the prostate is inflamed. We're not necessarily sure why. I'll give you some ideas as I move through the talk. But what you see are these pockets of inflammatory cells here. So there's chronic inflammation, which is very common. This particular slide has a little area in the lumen that's full of neutrophils, which is indicative of acute inflammation. And these lesions here called atrophy, which Angelo has described as a precursor lesion, which I'll tell you a little bit about. So this is not normal prostate. This is very different. This is just another view of what inflamed prostate looks like. Again, I'm showing you the atrophy, these lymphocytes, these chronic inflammatory cells. I won't talk today about corpora amylacia, but I love talking about corpora amylacia, so if anybody wants to hear about them, please come find me. We think that they're related to infections and inflammation in the prostate, and again, something that's very commonly seen in the prostate of the adult male. And so this atrophy that I've alluded to is some of the atrophy is characterized as something that Angelo has described called proliferative inflammatory atrophy, or PIA. And when you think of the term atrophy, you might think of cells that are dying or dead or quiet, and that's really not the case here. That's not what proliferative inflammatory atrophy is. What you're looking at over here is an H&E stain of PIA. Again, these are the atrophy lesions. There's some corpora amylacia here, and then there's chronic inflammation. There's lymphocytes scattered throughout the stroma all throughout this picture. And then over here on the right-hand side is a immunostain for Ki67, which is a proliferation marker. And so all these brown dots that you see are epithelial cells that are actively proliferating in this atrophy lesion. So this is not a quiet lesion. And furthermore, we're starting to study this a little bit more. I just wanted to show you this as one example and highlight Corey Porter, because I'm going to highlight all my students because they're here. So Corey's a graduate student in the lab. And just tell you that she's studying this one molecule called Lactoferrin, which is really intriguing. I won't talk about it more than to say that it's an acute inflammatory protein. It's contained in neutrophil granules, and it's related to bacterial infections, and it's highly, highly upregulated in areas of atrophy. So all this brown that you see here are these cells just pumping out this lactoferrin, and this is not normal. This is normal prostate epithelium that doesn't have any lactoferrin production, but is very high. So these atrophy cells are not quiet. They're proliferating, they're secreting things that aren't normally secreted in normal prostate epithelial cells, and they're very different. And then Angelo went on to describe that if you look very closely at histology of the prostate and men that have prostate cancer, you can actually find these lesions where prostate atrophy, which is right here, is directly merging. This is the same gland here, directly merging with what is thought to be the direct precursor lesion to prostate cancer development, or PIN, prostate intraepithelial neoplasia. So this is the working model. I give total credit to Angelo and Bill Nelson. This is their working model. It's just something that I study of the different stages of the earlier stages of prostate cancer progression, whereby one of the intermediate stages is PIA, or inflammatory atrophy, which is inflammation related, and it shares some of the molecular changes that you see, and the direct precursor, which is PIN, that we think directly is involved in development of invasive cancer. So why? Why is there all this inflammation in the prostate? What are some of the possible reasons for this? And so I'll talk largely today about infectious agents. That's what my lab studies. We study prostate infectious prostatitis as a potential stimulus for this inflammation that's so common in the prostate. But there's several other factors that have been described, including hormonal changes, estrogens. Again, Don Coffey has beautiful work on this. Physical trauma, including these corpora malacia that I told you about, urine reflux into the prostate, which is something that we're very interested in terms of the microbiome. I'll tell you a little bit about that. But if urine is refluxing into the prostate and there's actually a flora in the urinary tract, then along with the urine is flora, or bacteria, or other microorganisms being washed into the prostate, and this can happen in men. And then I'll tell you a little bit about one dietary factor, which is PHIP, which is a dietary carcinogen, but there's other dietary factors that can lead to inflammation in the prostate. And in fact, everything that's listed here in this nice graph that is published again in a paper by Angelo and Bill, a wonderful review article if you're interested in the topic and want to read up on it. It's a Nature Reviews article. But all of these factors, I added a few more: autoimmunity, and then the whole idea of the aging prostate and the microenvironment, the stromal components, that as aging occurs, these things tend to promiscuously start to secrete cytokines and other factors that might be pro-inflammatory. So all of these things have been shown in animal models to cause prostatitis. So I'll be narrowing down in on infections today, but there's lots of things that could contribute. Okay, so I told you I'm going to talk about infections in prostate cancer. We started to try to model this in mice. I thought I would highlight Debika here, even though she's moved on to medical school now, but I know that she is a graduate of this department. And this is some very nice work that she did developing a mouse model of P. acnes-induced prostatitis. So P. acnes is Propionibacterium acnes. It's a bacterium that you might or might not have heard of, but it causes acne, which is where it gets its name from. So it's a very pro-inflammatory bacterium, and there's strains that are specific to the skin, but then there's also strains that are known to reside in the urinary tract. And in fact, when we try to culture bacteria from prostate tissue from men that have had their prostates removed for treatment of prostate cancer, we can culture P. acnes. And what we've done is we've now taken some of these strains that we've cultured from humans and inoculated them back into mice to see what would happen. And this study that was published by Debika is one of these strains. And this is just the model of what she did. So what happens is early in the animal's life, we use mice that are about 8 to 10 weeks of age. You introduce P. acnes into the mouse prostate via urethral catheterization. And yes, that is as difficult as it sounds, but it can be done. So you inoculate via the urethra the P. acnes into the prostate, and then you wait for a very long period of time. It's just one dose, and we've taken the model out to at least a year so far. And what Debika showed in this model is that it takes a while for the P. acnes to get going. If it was something like E. coli, normally that would induce inflammation within 24 hours. With P. acnes, it takes a little bit longer. It takes about a week. And we think that that might be related to an intracellular infection that, again, in the interest of time, I can't tell you about today, but can talk to you about if you're interested in. By 2 weeks' time in this model, so early on there's chronic inflammation, acute inflammation, so there's neutrophils in the lumens of the glands. By 2 weeks there's raging chronic inflammation, a very pro-inflammatory bacterium. But even 8 weeks after the bacterium has been introduced into the prostate, and now we've taken this model out to a year, and shown that even a year after you've inoculated this bacterium into the prostate, you have a decent amount of chronic inflammation that persists in the mouse prostate. And if you stain or you try to go in and find P. acnes in the prostate a year after inoculation, you can't find it. So either there's very low levels, or it's formed a biofilm or something, or there's some reason why we can't see it, or this chronic inflammation is persisting without high numbers of bacterial cells still present in the prostate. We're not sure why that is, and that's something that we're following up on, but the phenomenon does seem to require live bacteria, because if you do the same thing with dead P. acnes, you don't get the same long-term response. And if you'll harken back to that picture that I showed you of proliferative inflammatory atrophy in the human prostate, this is also a Ki67 stain, where we show that at 2 weeks after infection, there's a lot of proliferation going on. Some of these are inflammatory cells, but even in the epithelial cells, there's a lot of proliferation. But again, this proliferation, even of the epithelial cells, seems to persist. This is 8 weeks that I'm showing you here after an infection with P. acnes. There's some other things that we stain for. NKX 3.1, which is thought to be a tumor suppressor, is decreased in this in these mice in the areas of inflammation. The same thing happens with androgen receptor. This has been described before in other animal models of bacterial-induced prostatitis, one that used E. coli that was published by Chuck Biebrick and Angelo. Chuck Biebrick is at UMBC. So this is a common theme with lots of the prostatitis models. And these are a lot of words on this slide here, so I don't expect you to read every single word on it, but I wanted to make the point that there are a lot of different bacteria-induced prostatitis models in rodents, and they all seem to have a similar theme. So you can use chlamydia, you can use E. coli, you can use P. acnes. We've done some studies with E. coli, both in rat and mice, and the consistent thing is that you can use a single infection, you can induce very long-term inflammation. It doesn't seem to require the persistence of the microbe itself. And there's multiple different types of organisms that can do this, but all of them are very pro-inflammatory. They disrupt the architecture of the prostate. So what did we learn from all of this up until this point? So what's the point of me telling you all of these, little tidbits about animal models is that basically, I started out by saying we think that inflammation is important in cancer development. So we can induce inflammation in the mouse prostate and rat prostate. We can keep it there for the animal's entire life, but inflammation alone, at least the type of inflammation that we're inducing with bacteria, does not seem to cause cancer. This does not happen in any of these models. To my knowledge, there's no model of mouse prostatitis caused by a bacterium that after some period of time induces cancer in a rodent. So then we started thinking, and again, this was about the time when I was a postdoctoral fellow with Angelo, about could we combine, so we think inflammation is important. It's been shown in other types of cancer that chronic inflammation is a bad thing. It can help to drive or promote cancer development or progression. So we think it's important in prostate cancer, but it doesn't seem to do anything by itself in these infection models in rodents. And so Angelo was studying this dietary carcinogen called PHIP, which is a mutagen. And we were thinking about it, talking about it, and the idea came about, well, why don't we see if there's a synergistic interaction between two different exposures, and one of which is a mutagen, the other one which induces inflammation, and see if that seems to alter the biology or drive cancer in this model. So I'm going to tell you a little bit about PHIP and a little bit about the PHIP model. And I know that there are people in the room that know a lot more about PHIP than I do, but just to give you a broad overview, PHIP is a heterocyclic amine. It's produced when you cook meat at high temperatures, and this is any kind of meat, beef, chicken, pork, fish, it doesn't matter. PHIP can be produced depending on how much you cook it and the temperature that you cook it at. So PHIP has to undergo activation in the liver, specifically by cytochrome P450 CYP1A2. But once it goes activation, what it can do is it can adduct to DNA and adduct specifically to guanine nucleotides, and it can cause G to T transversion. So PHIP can be a mutagen we think in humans, but also in rodents, and specifically in rats. So this has been linked, there are plenty of epi studies on this in human beings, and it seems to be linked to multiple different types of cancer, including prostate, colon, and breast. And in fact, when you give it to rats, it induces those types of tumors, small intestinal, prostate, and breast. And then there's also some blood tumors, some leukemias that can occur from PHIP consumption. So I'm going to tell you a little bit about the model that we have of studying PHIP in rats. So specifically, we study it in rats because they possess the correct cytochrome P450 to activate PHIP. I will not tell you about trying to translate this into a mouse model, which we've been putting a lot of work into, but today I'm just going to talk about the PHIP rat model. And so this is what happens when you give a rat PHIP via its diet, either gavage or in the diet. for a long period of time. This particular model that I'm showing you here is PHIP consumption, PHIP in the diet at 200 parts per million that the rats eat for 20 weeks of time. And I'll show you a timeline of that here in a moment. But I just want to start again by showing you a histology picture of what the tumors look like. And I call them a tumor. But what actually happens when you give a rat PHIP, what happens in the prostate, is they develop these cribiform pin intraductal types of lesions. So this is not an invasive cancer. It's a precursor cancer. It could also be classified as a carcinoma in situ, but because it is not invasive, it's not truly what you would call an intraductal tumor in human beings. So for lack of what a better term to call these in the rats, what we call them is cribiform pin / carcinoma in situ. So you'll see on my slides, it'll say cribiform pin / CIS. But those are these precursor lesions, neoplastic lesions that we see in the rats. And interestingly, I'll tell you a little bit about this. The cancers that the rats get are confined to one lobe of the rat prostate. So the rat prostate, similar to the human prostate, even though anatomically it's a little bit different, has different lobes. So the rat, there's a ventral, there's an anterior, there's a lateral, and a dorsal lobe. In the human, there's different zones of the prostate. There's a transition zone, which is where BPH forms. There's the peripheral zone where prostate cancer typically forms. There's only one lobe of the human prostate, whereas there's several anatomically separate lobes of the mouse prostate, but it's similar. This similar theme only forms in the ventral lobe of the rat prostate, only forms in the peripheral zone of the human prostate. So there's something to that, we think. And so this is a study that Angelo did that I'm just going to tell you as kind of a lead into what I'm going to tell you about what happened in the PHIP study that we did when we combined it with infection. So what they did is they looked at PHIP consumption in what's called the big blue rats, which are rats where you can basically calculate the mutation frequency in certain tissues that occur when you introduce a carcinogen. And what they showed, the take-home point from this paper is that all of the lobes of the rat prostate and other organs, even some organs that don't get cancer when you give a rat PHIP, all of them get mutations. And it's almost an equal level. There's an equal number of mutations in all the lobes of the rat prostate. So it's not just mutation alone that it can account for this ventral lobe specificity of the tumors that develop. And what they showed in this paper was that specifically in the ventral lobe, and so Angelo has always been thinking about inflammation. They thought about that in the context of this study too. There's no overt inflammation that forms when a rat eats PHIP. So there's not this, really severe chronic inflammation that I showed you about in some of the other pictures, but there was a subtle influx of inflammatory cells. What Angelo noticed is that those inflammatory cells seemed to be mast cells and macrophages that were coming into specifically the ventral lobe of the prostate. And what they did was they went in and counted these things and showed that indeed, just specifically in the ventral lobe, there was a marked increase in mast cell number and also in macrophage number. So, what they concluded, which doesn't seem to have made my slide for some reason, but what they concluded was that PHIP can act as both a prostate cancer initiator via mutation and as a prostate cancer promoter via the induction of the subtle inflammation that occurs specifically in the ventral lobe of these animals. So taking the next step in the study that we did, we hypothesized, okay, so there's this subtle increase in inflammatory cells that occur with just PHIP. There's these pre-invasive lesions that don't seem to become invasive for some reason in the rats. Well, what would happen now if we take rats that are given PHIP and causing the mutations in the ventral lobe and then inflame the heck out of them by introducing a bacterium as well? Could that overt, very severe inflammation drive the tumor to be even worse? And so that was the rationale for this study. And for this particular study, we didn't use P. acnes, we used E. coli. But it doesn't seem to matter. So we used this strain of E. coli, again, that was isolated from a human being. It was isolated from a man who had chronic prostatitis, chronic pelvic pain, so it's a clinically relevant strain of E. coli that we were using in this rat model. Same thing that we saw with P. acnes, where we give one dose of E. coli, they get raging. This is neutrophils again. This is acute inflammation, very severe chronic inflammation. in early time points, and it peters off a little bit, but it's still very severe inflammation that lasts throughout the animal's life. And this is the outline of the study that we did. We had a controlled group that just got control diet. This is AIN 93G diet. The PHIP alone group that got PHIP in the diet, mixed in with the diet at 200 parts per million for the first 20 weeks of the study. So it's a PHIP alone group. And what we did was we wanted to make sure that all of the animals were equally exposed to PHIP. So we weighed the animals and we weighed the diet, and we made sure that there was no difference in the amount of exposure and the amount that the animals were eating for PHIP. Then there was an E. coli alone group. So this group here was on controlled diet. They got 1 dose of E. coli 10 weeks into the study. And then we combined them. So PHIP for the first 20 weeks, and then 10 weeks into eating PHIP, they also got dosed with E. coli. Then all animals for the last 21 to 52 weeks of the study were just on controlled diet. So our goal was to look at the prostates of these animals and see what happened with the prostate lesions. What happened was that we were completely surprised and that all of our animals that were in the PHIP plus E. coli group died early. They started dying off very rapidly. And actually, this was not due to prostate cancer. And it just kind of took us by surprise. So this is a survival curve. The PHIP plus E. coli group here is here. There were some animals in the PHIP group that had to be euthanized because of benign tumors that got too large for the animal guidelines. So this over here is a survival curve where we're just looking at cause-specific survival, so the animals that died of invasive cancers. So the PHIP plus E. coli treated animals died earlier, and they died because they developed twice as many invasive tumors in areas they developed the prostate lesions, but they didn't die of them. But they developed very bad cancers in other areas, including the skin, the small intestines, and the Zimbel gland, which is a gland in the ear of the rats. And so this was very curious to us. There seemed to be some sort of systemic effect of infecting the prostate and inducing this raging inflammation in the prostate, and the effects of the immunogenic effects of PHIP at other areas in the body. So the next thing we did was we did focus on the prostate for what we could. So the PHIP plus E. coli animals, remember, they died much earlier than the PHIP alone treated animals, so this is not an age-matched study. We really weren't expecting to see an increase in the number of lesions or the size of the lesions in the prostate just because the animals were much younger and the combined group. There was a subtle increase in the number of lesions in the PHIP plus E. coli group. But interestingly, so this is what the tumors look like. And then we measured Ki-67, we did a Ki-67 stain and measured the number of Ki-67 positive stain and showed that there in the tumors of the PHIP plus E. coli treated animals, there was a significant increase in the number of Ki-67 positive cells in the tumor, indicating that if they had lived long enough, maybe we would have seen a difference. Don't really know. Also, interestingly, we quantified inflammation in this model and showed that specifically in the ventral lobe, there seemed to be a prevalence of chronic inflammation, both this is the E. coli alone treated group and also in the PHIP plus E. coli treated group. So there was acute and chronic inflammation present in the other lobes as well. These are animals at end of study, either when they died or when they made it out to the full 52 weeks. But again, this curious predilection of the ventral lobe specifically for the increased amount of inflammation. And then I also wanted to highlight that, as I told you about an Angelo study, he quantified mast cells. Heidi Hempel, who's also sitting right in the front, another graduate student in the lab, has a beautiful thesis project on mast cells. Absolutely go talk to her if you're interested in mast cells. She'll tell you all about mast cells and why they are causing human prostate cancer. But for the relevance of this study, what she did was she quantified the mast cells in the prostates of these animals at time of death. And again, so Angelo study quantified mast cells 8 weeks after PHIP exposure. We did some canary animals where we looked at them 20 weeks after PHIP, so the 20 weeks of PHIP, so directly after they were done taking the PHIP. And there was this increase in the number of mast cells in PHIP-treated animals. But even 52 weeks after the animals had stopped eating PHIP, so I told you after 20 weeks they go on control diet and stay on that diet for the rest of the study. even 52 weeks or an end of study, there's still this elevation of mast cells in PHIP alone treated animals that's significantly different from control, and they're even higher in E. coli treated animals and PHIP plus E. coli treated animals. So that was interesting to us. So again, there's something that seems to be altering fundamentally the biology of the ventral lobe of the prostate in these animals that are exposed to PHIP, and it's even worse when you infect it. Same thing with macrophages, although we didn't quantify them, but you can see they're increased in number in PHIP, E. coli, and PHIP plus E. coli. All right, so then we started thinking, well, why were all these other tumors so bad when we thought we were inducing inflammation specifically in the prostate and that we were going to drive worse prostate cancer. So one of the things we thought to do was to look systemically, to look at any sort of cytokines that might be induced in the serum of these animals that are, correlated with the response to infection in the prostate of these guys. But this has been described not well, but to some degree in the literature, that circulating cytokine levels could have an influence on tumor development and progression at other sites. And so what we did was we looked at the serum of the animals and we looked at a panel of cytokines. But what seemed to fall out as the ones that were interesting were IL-6, which is elevated both in PHIP alone treated animals and even higher in PHIP plus E. coli treated animals, and also IL-12, which was a little bit up in the E. coli treated animals, but then also increased in PHIP plus E. coli treated animals. Nothing else seemed to really pan out with the exception of leptin, which was decreased in animals that were given PHIP and PHIP plus E. coli, but leptin is associated with body size in the animals, and when the animals start getting sick, when they start developing tumors, they start to lose weight, so we think that that might explain some of that decrease. When you look at the circulating cytokine levels in correlation to whether or not the animals developed prostate lesions, interestingly, we found a significant correlation between IL-6, circulating IL-6 levels, and the incidence of these ventral prostate tumors. We did not see that association for IL-12. That's interesting to us in that IL-6 and systemic levels of IL-6 have been extensively described in the literature as being correlated to advanced prostate cancer. And I had another graduate student in the lab, Xu Han, who recently graduated, who her entire thesis was looking at IL-6. And I just wanted to make the point here that we seem to be seeing another theme coming out of all of this, and that when we looked at, so one idea is that if there's IL-6 being elevated in the serum, maybe it's coming from the prostate tumors themselves. Maybe they're pumping out IL-6 and somehow that's getting into the serum. But when we did in situ hybridization for IL-6 in the tumors themselves do not seem to make IL-6. When you have areas of inflammation, specifically areas of acute inflammation, you can have a lot of IL-6 upregulation. It's not necessarily by epithelial cells, it's by the inflammatory cells. And I just wanted to make the point that Xu Han did this same study in human beings and found the same thing in human beings, where the tumors never make IL-6, but when you have inflammation, specifically acute inflammation, it's highly upregulated. But in animal models that she did with xenograft studies that I'm not going to tell you about today, there does seem to be this interesting correlation between systemic levels of IL-6 and tumor growth. So we're not really sure what that means, but it's certainly something that we'll be following up on in the future. So to summarize this first half of my talk, what I've told you about so far, some of the key points is that a single infection in the prostate seems to set up this chronic inflammatory event that persists for a very long time. We're not exactly sure why that happens, but it happens with several different strains of bacteria. It seems to be specific to one lobe of the rat prostate. And in the mice, I didn't mention this, but it's also specific to one lobe, but a different lobe. It's actually the dorsal prostate in mice. When rats were fed PHIP concurrently with this infection from E. coli, they seemed to die quicker, and this seemed to be not because of, it was not because of prostate tumors, but because of tumors that developed in other sites more rapidly. And we think that this might be due to some sort of systemic phenomenon that we don't fully understand yet. To our knowledge, this is the first documented case of an infection that we think was limited to the prostate, although we haven't proven that the E. coli cells didn't leave the prostate and end up in some of these other sites. It's certainly a possibility. As far as we know, it's the first documented occurrence of infection or prostatitis leading to an acceleration of carcinogenesis in other sites of the body. Could this be mediated by systemic cytokine levels? Maybe. And in fact, if you look in the literature, you dig deep, there are a few studies studying Helicobacter hepaticus in animal models and showing that infection of the gut seems to drive systemic cytokines. And interestingly, one of the important ones there seems to be IL-12, and that seems to influence tumor development at other sites than the gut, including both the liver and the mammary tumors. So those are the key points from what I've told you up until this point, and I thought the whole H. hepaticus in the gut was actually a good segue into the next half of my talk, which is going to be about the microbiome. And I promise you, I think they are all related, that these two halves of my talk are not separate from each other, but what we've been thinking about recently in the context of the microbiome or the metagenome is, could there be a potential source of these prostate infections that we think are important in men living in the urinary tract? So what we're learning from the Human Microbiome Project and from all of the data that have come out of these recent microbiome studies is that basically every part of the body that is exposed to air is colonized by microorganisms. This is really well described for places like the gut, where there's order of magnitude more microbial cells than there are human cells in the body. But this is now recently being described for other areas, including the skin. It's very well described for the vaginal flora in females, but the Human Microbiome Project completely disregarded the urogenital. Aside from the vagina, they did not pay any attention to the urogenital tract in their initial study. They're starting to get on board now when they're looking at the association between the microbiome and disease, because there have been some papers published saying, wait a minute, there might be a urinary tract flora, but there's way less known about this than the other sites that were included in the Human Microbiome Project. So I'm starting to think about how the microbiome could relate to prostate cancer. What I'll tell you about here in the last half of my talk is going to be exclusively on the urinary microbiome. So if you know your prostate anatomy, again, I've talked about some of these things. This is just a cartoon of the prostate. I've mentioned transition zone, which is this zone of the prostate where BPH forms. And it is right around where the prostatic urethra is. So the urethra runs through the prostate. The peripheral zone is out here. This is where most prostate cancer develops. But the point that I'm trying to make is that the urethra runs through the prostate, and we think that there is a flora in the urethra, and we don't know anything about it, but it could potentially either influence prostate infections or serve as a source for prostate infections. So we're trying to start to study this big time in the lab. So those are direct interactions between the microbiome and the prostate that we're interested in. And there's a ton of indirect interactions that I won't talk about today. I hate to disappoint, I'm not going to talk about gut flora. But I just list a few here that could be indirect interactions between the microbiome and prostate cancer, including the microbiome's influence on obesity, which has been linked to prostate cancer, different dietary carcinogens, including PHIP. So we're starting to study the gut flora's influence on metabolism of PHIP, which is something that, as far as I know, has not been looked at before. But even things like dietary soy that is metabolized by bacteria in the gut. So soy is supposed to be anti-inflammatory. That might play a role in prostate cancer, but it all depends on activation by certain microbes in the gut, or metabolism by certain microbes in the gut. And then if you've seen any issue of Science recently, you've seen a paper in there, they're trickling out like every other week now, showing that the microbiome in the gut can have a profound influence on certain cancer treatments. And this has been described very extensively for immunotherapy. So these are all things that we're very interested in starting to study and starting to get a handle on, but today I'm going to talk about urinary microbiome. So we'll start with the dogma. So the dogma is that urine is sterile. And I know that clinicians think this, because whenever I've gone to talk to a clinician about the urinary microbiome, they look at me in the eye, Karen, urine is sterile. What are you talking about? And the truth is, actually, it's not. And there's lots of reasons that we should have known that it's not. One of which is because even when you send a sample out for urine culture, it's not that it's positive or negative, there's a threshold value. So things grow on the plate. It's just if they don't grow on a plate above a certain threshold, you don't consider it an infection in the urinary tract. But the problem is that all of these microbiome types of studies have been exclusively dependent on whether or not you can culture a microbe. And the truth is you can't culture most of the bacterial species that are out there. And so now that we have culture-independent types of methods, such as sequencing and sequencing of the 16S gene, which I thought I had a slide on next, but I guess I didn't, so I'll tell you a little bit more about it in a moment. So now that we have culture-independent ways of profiling microbial communities, we're starting to find out that there's rich flora in areas that we didn't previously think that there were. And so, like I said, the urinary flora is something that hasn't been as well studied. However, there are a collection of studies and even a review article that just recently came out in Nature Reviews Urology, if you're interested in going to take a look, that have started to look at urine microbiota in both men and women. It's been predominantly done in women, and I think just because the people that kind of got into this happen to be the same people that were studying female related urinary tract disorders. But there are a couple of studies in men. We're hoping to increase that quite a bit. So some of the interesting things that have come out of these studies so far, I'll just give you a few little vignettes to see if I can get you interested and see if I can get you convinced that this is actually a thing. So one of these studies was to look at, this is adolescent males, and what they're looking at is the difference in flora in either the penile coronal sulcus, so the outside of the penis, versus the distal urethra. So the point of this study was basically to convince you that the bacteria that you see in the urine and in the urethra is actually different, and not just contamination from the skin outside of the urinary tract. I mean, you don't even look at the taxa on here, just look left colors, right colors, it's different, okay? That's the point of this. So if you profile both, there's shared species for sure, and certainly the skin around the urethra can influence what's in the urethra, but they're not the same, it's something different. They've also shown that if you match urine samples to a urethral swab, you get the same answer. So urine samples can be used to study urinary flora. There's some literature out there indicating that unlike the gut, where if you were to profile at the phylum level, just take a step back to the phylum level of bacteria and look at gut flora amongst many individuals, men and women, you see a very similar profile. The gut is dominated by certain phyla of bacteria. That's not the same for what we know so far about the urinary tract. There seems to be groupings of different types of, there's still some dominant flora. Again, this was all done in women, but groupings into different urotypes. So there's a rich flora in the urinary tract, but it's not constant between individuals. And there are some intriguing studies that have just come out looking at dysbiosis in the urinary flora and how this could relate to infections in the urinary tract. So same principle that you would use in the gut, for example, that where you have this normal healthy flora, it's very diverse, and once you start to alter that flora for whatever reason, it could be something in the diet, cancer that's forming, there could be multiple reasons why you could get an infection, alterations in the normal level of diversity, and that seems to lead to pathogen outgrowth, right? So this has been shown now, this exact principle has been shown for urinary tract infections, where men and women that tend to get urinary tract infections tend to have less diversity in their urinary flora for whatever reason. And interestingly, there was this study done by this guy, David Nelson, up at Indiana, where they looked at asymptomatic men. So these were men that were going to an STI clinic, either as they were there with somebody else or they were there for not symptoms of an STI. Some of these men were found to be carriers of certain types of STIs. And those men that were carriers of these STIs also seemed to have overgrowth of a certain type of organism in their urinary tract. So this kind of blows my mind even just to think about it, but it makes sense, and I think that there's going to be some exciting data coming out of this in the future. So in the last few minutes here, I'm just going to tell you about a project that we started. It was funded by the V Foundation, so this whole idea of whether dysbiosis in the urinary tract. So we study prostate infections. We think the infections lead to inflammation. We think that inflammation is bad in terms of prostate cancer development. So these are a lot of words on this slide, but the point is we wanted to start studying urinary tract flora in men with and without prostate cancer and trying to understand, is there a difference in the fundamental profiles of their urinary flora. oh, here's my slide on the strategies for studying the different sequencing methods. What I'm going to tell you about today is simply sequencing of the 16S rRNA gene. You amplify a certain fragment of the gene and you sequence those amplicons and use that to profile all different bacterial species. It's limited to bacteria, but it profiles all of the bacteria in your sample. And it's also the fastest and cheapest method of profiling a certain site. Metagenomic sequencing is sequencing all of the DNA in the sample. This will capture things like viruses, fungus, protozoa. It can also be used for functional analyses. We've done some of this. I'm not going to talk about it today. And then microbial transcriptomics or metatranscriptomics can be used to see what these things are expressing. Right. So the study that we did, this is what we've done to date as far as this study in urine specimens. We were looking at urine specimens as part of Dr. Alan Parton, who's the chair of urology, his bank of urine specimens that they've gotten from men that were coming here to Hopkins for some sort of indication for prostate cancer. So keep that in mind. All of these men had some sort of reason to be assessed for whether or not they had prostate cancer, either an abnormal DRE or an elevated PSA. And all of the specimens were taken, the urine specimens were taken post-DRE. And if you don't know what a DRE is, I don't necessarily want to describe it to you all right now, but let's just call it prostate massage, okay? So all the men underwent so the urologist is feeling the prostate, feeling for prostate cancer, but in the process, the prostate gets massaged. And so we were thinking, well, maybe that will enrich these urine samples for any microbes that might be in the prostate as well. We did not do pre and post DRE, and we absolutely need to do as a follow-up study, a group of men that didn't have a DRE before. But as a starting point, we wanted them all to get DRE. We wanted it to be enriched for anything in the prostate. We looked at pelleted urine. We've done 134 of these samples to date. 64 were from men that were found to have cancer at a biopsy that was taken after they gave the urine sample. 65 were controls, meaning they were biopsy negative. And then 5 of the men that we've captured to date actually later went on to develop or to find cancer. So we kind of separated them out because they weren't a true negative at the time, but we knew that they went on to be detected to have cancer. We made sure that they had no prior biopsy, or at least if they did, it was greater than a year since their prior biopsy because it's a transrectal biopsy. Basically, the prostate gets flushed with rectal flora when the biopsy occurs, and we didn't want that to influence anything we found in the urine. And we wanted to make sure they weren't on antibiotics at the time of collection. And we did a bunch of mock extractions as negative controls. So I just told you all of this. We looked at urine pellets. I could spend an entire seminar telling you about the extraction protocol that we came up with for DNA extraction. It took several years of a predoctoral student's life to settle on a method that we're very happy with, that we're getting lots of DNA from both gram-positive and gram-negatives. We did our 16S amplicon sequencing, and I'll tell you a little bit about a real-time PCR assay that we developed for bacterial load in these samples. So this is what they look like when you amplify 16S ribosomal rDNA from bacteria from urine pellet samples. And what you can see here is that just running it on a gel, you can already tell that there seems to be a difference in the quantity of bacteria in these different urine samples. Some are very strong, some not so much, and we developed a real-time assay for this, but this is very similar to what's been seen in other studies as far as bacterial load. And just to show you more so a pretty picture, more than anything else, of what these profiles look like in the men without cancer and the men with cancer. This is at the phylum level, so again, looking very bird's eye view of what's going on. Really, we just got this data back, we're starting to mine through it, we're looking at all the species, there's tons of species that are present in all of these samples, and really I think when we're looking at differences, the things that we're going to be looking at are the things that are kind of down here in the trenches. But some of the things that surprised us that we weren't expecting is all of this red in a lot of the samples. So again, it seems to be grouping into urotypes, but this is very different from what's been described in women. So in the men, a lot of the men seem to be dominated by actinobacteria. And interestingly, the P. acnes, the Propironibacterium acnes that I told you about in the beginning of my talk is a member of the actinobacteria, and in fact, P. acnes seems to be enriched in the urinary tract from men. This is not found in women. P. acnes is not a major component of the urinary tract in women. So we're interested in that. It's an intriguing finding. And then I'll just finish up here in the last few minutes telling you about the quantification of bacterial load in these samples, just because I wanted to highlight one other student who's sitting in the front row here, which is Eva Shrestha. So she just joined the lab, but has already got data that I'm presenting at a seminar, so that's great. So she developed this real-time PCR assay for quantifying the numbers of bacteria in these samples. This is benign versus cancer. It's kind of interesting. There seem to be fewer numbers of bacteria in the cancer group, and thankfully in our negative controls, which is a blank extraction that we take all the way from DNA extraction through to sequencing, doesn't seem to have much in it, so that's good. And then just to leave you with a little intriguing piece of data that we absolutely need to follow up on before I can say anything definitive, but when the biopsy results are reported, the Department of Urology, when they are recording the data in their database for the urine samples, they will report the most severe lesion reported on the biopsy results. So if they have cancer, they write down cancer. If they have PIN, but no cancer, they'll write down PIN. Inflammation is the next one, and then atrophy and benign. And so what we saw is that when the pathologist actually recorded that there was inflammation in the biopsy, that seemed to track with the number of bacterial cells that we found in the urine. So the problem with that is there's a couple problems with that. Number one, the pathologist might have seen inflammation and didn't record it, so it didn't get put onto the biopsy result. Or, some of the ones with higher lesions might have had inflammation, but because they had something worse, that's what was recorded. So there's a lot of reasons why we need to follow up on this, but just as an initial interesting finding in all of this, the ones where they did actually write inflammation seem to correlate to the bacterial load in the urine. So in summary, point one, there is a urinary microbiome. Hopefully I've started to convince you that that is a thing. It's very diverse, and there's substantial variation between individuals. Also between males and females, which we think is interesting, and probably also between different age groups. I didn't talk about it at all, but there are studies starting to be done on this, that adolescent men have a very different urinary tract microbiome than adult men. And in fact, after males start having sex, the urinary microbiome is altered, and it starts taking on flora from, it makes sense, the female vagina. But so once men start having sex, their urinary flora is altered. It starts taking on some of these species that are more prevalent in women, but that is very stable. So the colony or the diversity that seems to populate then seems to be maintained throughout the rest of your life. So that's extremely interesting biology that we're just now starting to understand. So loss of diversity seems to be associated with genitourinary disease. We think it's associated with prostate cancer, prostatitis, but there's also literature to support this in regards to urinary tract infections, urethritis, and STIs, and possibly also urinary incontinence, which is some of the data that I showed you in women. And we don't know yet if it's related to prostate disease. But the bacterial load seems to vary between individuals, but we have some intriguing data to indicate that it might correlate to the level of inflammation, at least in the prostate. There's a ton of future directions, some of which I've already mentioned, metagenomic sequencing to look at other things in addition to bacteria, looking for men without indication for prostate cancer, such as men that weren't being seen because they were suspected to have prostate cancer, longitudinal studies to see if anything we think is important predated the development of cancer in men, and then the pre versus post DRE samples that I told you about. This could also be correlated to a bunch of different environmental types of factors, other types of factors, including race, diet, geography. And one thing that that Eva is working very hard on right now is developing this in situ hybridization probe to go into the tissues now and look to see if the things that we see in the urine are actually in the prostate and associated with inflammatory lesions there. So with that, I will end and acknowledge the people that actually did the work. Again, I have to highlight Angelo and Dr. Nelson, who certainly, I just took the next step. They did everything before that. Kirsty and Brian were heavily involved in the PHIP studies. I told you all about my students because I love them and I love to talk about them. Vasan Yagnisubramanian is one of our colleagues. He runs the Next Gen Sequencing Center that's next door to our lab. So instead of sequencing cancer genomes, I've forced the poor guy to be sequencing bacterial genomes for me, but he seems to be okay with it. He's very helpful. And Sarah Whelan, who works with him as far as the bioinformatics. Leslie's Department of Urology, helping with the urine samples. Of course, Dr. Parton. Ted Schaefer helped with providing the strain of E. coli that we use for the PHIP study. Jessica Hicks is our lab manager. Without her, we would not be functioning. And then there were lots of different funding sources. Norma mentioned the Prostate Cancer Foundation, of course, which has been very beneficial to my career up until this point. Check us out on Twitter. Follow us on Facebook, and that's about it. Thank you.
We have a few minutes for questions.
Yes.
Thank you very much. I've been involved in looking at UTIs in women, and I think there's possibly a lot of information that can crosstalk that would be extremely useful. And to the extent that now we're realizing that men may have issues as well,
yes.
and urine may not be privileged in men, as we know it hasn't been
yes.
in females
yes.
for a long time. But one of the interesting, a lot of this work is in collaboration with my former student, Lance Price,
Okay.
who's characterized the urinary tract microbiome in females.
Mm-hmm.
But one of the interesting things is that in females, the urinary microbiome can be influenced by diet.
Mm-hmm.
And so that may be true to some extent, possibly different because of anatomic differences. And so it might connect your two themes.
Absolutely. Yeah.
And the other is that I think that you're right. You have to go beyond simple taxonomy because, as you know, in the study of UTIs in females, we're very concerned about specific strains, for
Right.
example, E. coli.
Yep.
And therefore, if you start getting that information, that might actually jumpstart hypotheses rather than continuing to do the taxonomic surveys.
Absolutely. I'm not sure that was a question, but I agree with everything that just came out of your mouth. Thank you. Yes, Jelani.
Great talk. I'm wondering, over time, in the mice that had the PHIP and E. coli, do you see, over time, if you let those mice go out, do you see an increase in immunosuppressive cytokines? Because your work looked at IL-12 and IL-6 and other pro-inflammatory cytokines.
Right.
Do you see a...
Yeah, we have not looked at that in that model, not in the rat model. We didn't try to quantify any like Tregs, suppressor cells. You know who you could talk to about that though is Brian Simons, because he was on that paper. He was the one that gave us the strain of E. coli that we used, and I know that he's done additional studies in mice, and I'm pretty sure that he's profiled additional immune cells. So you might want to ask him about that. Yes.
So in your humans that develop cancer, it seemed like maybe their problem was some immune deficiency, that they weren't clearing the bacteria from mounting in the appropriate immune.
Yep.
So if you look for other immune.
So in humans, we actually have looked at suppressor cells, and we know that the Tregs are in there. We have a couple of published papers on this. We know that CD8s can express PD-1 in the prostate, and there's this whole idea, and we haven't really studied it, mostly because I'm not really sure how to study it. Maybe Dr. Coffey can elaborate on this, but there's this whole idea that the prostate might be an immune privileged site. So it develops fully develops, if I understand correctly, after puberty. And so that's not an issue until you do something like induce raging inflammation in the prostate, where the epithelial barrier, the normal architecture starts to get severely disrupted. All these immune cells are coming in, and so in addition to responding to the bacterium, they might be exposed to all of these antigens in the prostate that maybe the immune system hasn't been educated to. It's a theory. I know a gentleman at University of Maryland, Rich Alexander, has studied a little bit of this, looking at circulating T cells to certain prostate-specific antigens, and you find low-level numbers of these, and then prostatitis or chronic pelvic pain syndrome. But aside of that, I don't know of any extensive
One of
studies.
the other things that happens with the immune system with aging is
Yeah.
that as you get older, your
Yeah.
not able to clear these infections.
Yeah, I agree. Yes. Oh, sorry. Let's go. I'm tempted to be like, no, Dr. Kaufman, really, no, but please.
So I really like the PHIP example because it was really nutrition, food, diet choices. But are there other environmental exposures that are on your radar that are not just food related with cooked meats? Are there other broader toxicants or exposures that you're aware of that are sort of crossing over into your future directions?
Right. I mean, I'll tell you, for me personally, it's a little bit different. I'm sure that there are other environmental toxins that maybe are being looked at. One thing to think about is things like estrogens and dietary components that can lead to increases in certain types of estrogens. One thing, this is not necessarily environmental exposure, but something that we're really interested in right now, and I kind of alluded to it at the beginning, is urine reflux. Urine reflux. So urine as a, so this happens when, again, with aging, that you can have things not be as strong as they used to be, and you can have things like urine reflux, it happen in the prostate. And so that urine in itself can be harsh and cause damage in the prostate, but then think of that in the context of also washing bacteria maybe into the prostate. So that's something we're really interested in. In fact, if you're super interested in that topic, later on today at 4 o'clock, I'll be talking at the symposium that they're having for the Synergy Awards over in Zayed, talking about, again, about urinary microbiome, but this time in relation to BPH, which is in the transition zone, which is literally, so it's another disease of the prostate. It's benign, but it, just as, and numbers of men affected, even greater than prostate cancer. And we think that of any places where this process is going on, this like constant introduction of the stimulus, cellular damage, regeneration, and the prostate is growing in this area. It actually looks like scar tissue. And so I didn't even talk about BPH today, but we're very interested in that as well as a potential.
Karen, you always give terrific talks with great science and beautiful thinking. This question you know most about, but I want to throw it out for the young people in the audience if I could. First of all, there's vast differences in prostate cancer in different countries. In rural China, it's extremely low, as you well know, and then when they go western or move to Hawaii or the United, it jumps up.
Yep.
Now, this doesn't sound like genetics right away. It sounds like something else like what you're talking about. Now, I know that Angelo and your group and all of them have looked at inflammation between people in Asia's prostate and people in the United States area, and there's a big difference.
Right.
Now, when they move here, does the inflammation go up in the people from Asia?
I don't know.
Okay.
Ivo,
The
do you
second
know?
thing is the tremendous high concentration of prostate cancer occurs in Norway and Sweden, much higher than the United States. And is there inflammation in Norway and Sweden?
Yeah, I mean, unfortunately, I don't. The person you need to talk to about this is Angelo, and he.
Yeah,
unfortunately, he's not here. I don't think,
Yes,
but
and
he probably has a study on this. I
that's
just
why
don't know.
I'm saying
Yeah.
I'm throwing this
Yeah.
out for the young people,
Yes,
not
yeah.
to challenge you.
Yeah.
Now, what in the world could do that? And of course, in Jamaica, it's very high too, a lot different than it is here. And people know about that.
And I don't know if inflammation's
Yes.
been actually scored in
Yes.
in these
so
different
we need
groups.
to look at the inflammation in these various migrant situations and in the various geography. You had some of that in your last question. So the question is, if that were true, is there anything in the diet that could account for that? Well, you have PHIP, and we know that black barbecue in the lines in the United States isn't the same as people in Asia who walk food. But there may be something else involved. And I'd like to throw that out for the audience to think about. And that is, there's a vast difference in the diet in people in China. And you never see a piece of cheese in a Chinese, Japanese, or Korean restaurant, ever, that I know of. You don't see milk, and you don't see ice cream. Now, when they come to this country, they say, oh, well, they have lactose intolerance. Oh, no. when we put out pizza in our lab meetings, it is consumed in massive quantities, people from those countries. Okay, so it's not something like we're poisoned to lactose. It might be, but it didn't stop anybody from eating our pizzas and when they jump in line. I just want to leave it out. Now, could it be in the pizza and the milk that could do this? Is one of the only compounds found in the diet, which is not made in you, and we've talked about this, is not made in you, comes from the physician health study out of Harvard, where 47,000 people were followed over 20 years with follow-ups. And one thing in the diet jumped out as being high and causing a 70% increase in fatal prostate cancers. As you know, it's cholic acid, which is the methyl donor, which you do not make in your body. Well, it comes from your diet. The source of that is milk, cheese, and eggs, okay? So I just want to leave that. I think that all of this, as you're beautifully presenting, is an interaction between the inflammation, the microbiome, promotion, and things, PHIPs, and all this. And I want to salute you for doing what I think is, from what I've seen in this field, the most paramount work in this group. And so, thank you. I didn't mean to carry on, but I wanted to leave something for young people here to put into the machine in relation to what you're seeing.
Thank you.
Oh, by the way, only the dog of thousands of mammals, and all mammals have prostates, of thousands of mammal species, only one, no chimp, no gorilla, gets prostate cancer, and that's the dog. And it has inflammation of the prostate, which precedes, John Isaacs and Bill Isaacs, all of them did that, precedes the onset of an aging study in dogs.
Yep.
Thank you.
Thank you.
Thanks so
Thank you.
much, Karen.
Thank you.
Donovan, for wrapping it up. So, thank you.